Hi, everybody. Welcome to the first of 3 lectures that we have for Module 9. I'm going to go ahead and share my screen. We're going to first start out talking about viral infection and cancer. So this will be a short lecture just talking about the fact that we know that with a large percentage of cancers that they actually have a viral origin. So I'll go through this, and then the rest of this module will be covering the enteroviruses with a specific amount of time put on poliovirus. So let's start out with mini lecture 9.1, viral infection and cancer. So the learning objectives for this particular lecture should include the influence of virus infection on cancer. Also, you should know the characteristics of cancer cells in vivo. And that just means in a living person. We'll look at that and get some idea of what those characteristics of cancer cells are after we've already defined cancer. We'll also talk about the idea of cancer being a multi-step process, meaning that generally speaking, it's not just one thing that happens that causes cancer, but many things happening over a period of time that actually lead to cancer. We'll also talk about a new way of thinking about therapy for cancer called virotherapy. Some vocabulary for this particular lecture. There's quite a bit of vocabulary. I've put some of it up here, but I will walk through all of these as we move through this particular lecture. So first, to stop and talk a little bit about the history of cancer viruses and tumors. Cancer, we know, has afflicted humans throughout history. There's records going back hundreds of years showing that cancer existed. And what we do know is that about 20% of human cancers are associated with some sort of a viral infection, meaning that a viral infection either contributed or had something to do with those particular cancers. So the early research that was done was actually on a virus called the Rous sarcoma virus. In 1908, scientists first were able to see that there were things that we considered filterable agents, meaning that if we put a filter on a particular fluid, the filter didn't remove these particular infectious agents. And in this case, these ones produced tumors in chickens. We call them filterable agents, meaning that the filters at the time that we were using didn't have small enough holes that they could actually trap the viruses, so the virus would remain in the fluid as we put it through a filter. In 1911, Peyton Rous was the researcher who demonstrated that bacteria-free filtrate, meaning that bacteria are larger, so they weren't able to go through the filter, that the actual filtrate that removed the bacteria caused sarcomas in chickens. And so what that told Peyton was that it wasn't being caused by bacteria, but something that could actually make it through those filters. The agent was named the Rous sarcoma virus, and it's actually a retrovirus. And we'll talk more about retroviruses when we get into the chapter on HIV. When we think about cancer today, though, we can think of at least 6 viruses that are thought to contribute to about 20% of all cancers. So when you think of all cancers that we find in humans, there's 6 viruses that we think contribute to 20% of these particular cancers. And you may be familiar with some of these. We will be going into many of these in future modules, but I do want to at least recognize them here. So out of the hepatitis viruses, hepatitis B and hepatitis C, we know contribute to cancer. Human papillomavirus, which is one of the ones that's probably most closely studied, leads to cancer of the cervix. And so that's one that we know there's a vaccine that has come out recently. There's also the Epstein-Barr virus that we'll talk about in a couple of modules. This one has been shown to play a role in some of the cancers. And then some of the herpes viruses. So in this case, herpes virus 8 is associated with Kaposi sarcoma, which was a cancer that we saw commonly in early stages of AIDS patients. The last one then is these human T lymphotrophic virus types, and this is types 1 and 2. And these generally cause a leukemia, a problem with the T cells of the immune system. Some general terms, as I said, this particular module has a lot of vocabulary, so this would be a good place to spend time on studying this particular module, is maybe doing flashcards for this. So cancer is the term for any diseases where we have abnormal cells that divide without control. So basically what we're looking at is cells that go through cell division, but don't ever stop. They continue to divide, and that's where we end up with tumors. An oncogene, onco refers to cancer, gene meaning a gene in the genome. This is a gene that has the potential to convert a normal cell, whether that's in the skin or whether that's on the cervix, to a cancerous or transformed cell. So an oncogene is something that would be found in the cell that when it's actually turned on, it can cause that cell to become cancerous. We know too that there are what are called viral oncogenes. And so these are genes in the virus that are responsible for this virus causing cancer. And retroviruses can carry altered cellular genes that are tumor promoters. So basically, these retroviruses can bring these genes into human cells that will actually promote the growth of tumors. We also have, that we haven't really talked about, but if you've had a general biology course, you may have heard about this, something called tumor suppressor genes. And most of our cells in our body have these tumor suppressor genes that you can think of as kind of a safety net, that if there's a cell that's going through uncontrolled cellular division, that we would have what we call tumor suppressor genes that would be expressed that would stop it, that would basically tell that cell to stop. So these are genes that suppress or inhibit a normal cell from turning into a cancer cell. What we see, though, is with some of these oncoviruses is that those genes can be turned off or mutated to where they no longer work. And when these genes are turned off, they will cause cancer. So that safety net basically is removed. Cell transformation is when we have any change in the cell, and that can be what the cell looks like, so its morphology. It can be the way that it actually metabolizes nutrients, so biochemical, or it could be growth properties, the ability of this cell to grow or to go through cell division. And then one of our last terms here is metastasis. This is where when we have maybe a tumor growing, so this uncontrolled cell growth, that some of those cells break off and spread to a new location. And we call that metastasis. And that is where we really run into a problem of, instead of having it in one specific location, it's actually traveled through the body and is causing or spreading cancer throughout the body. When we think of cancer cells, then we want to think about the different characteristics that they would have. And these are what we see in vivo, meaning that if we were to look at cancer cells in the human body, we would see an increase in the mRNA, so expression of oncogenes, genes that cause uncontrolled growth. We would see increased genetic or gene expression, so more of those proteins that promote this. We would also see a loss of that tumor suppressor gene function. Remember, I said that's the safety net in our cells. We would often see that we would have a loss of that function. With those two things happening, we would expect to see increased or unregulated level of growth factors, meaning that the cell now is just going to be churning out growth factors, which means that it will divide uncontrollably. We'll get that cell going through cell division over and over and over again, often causing a tumor. We would also see increased levels of enzymes, and this would increase that ability to divide uncontrollably. And lastly, we would typically see these cancer cells being able to evade or escape the immune system. So our immune system would not do a great job of clearing any of these cancerous cells in the body. And that's because these are host cells that often they don't get recognized as something that's causing a problem because they have many of the markers of a host cell. So as I mentioned earlier, cancer we consider a multi-step process. And what I mean by that is that not just one thing happens to cause cancer, but it's multiple things happening in succession. And this is typically why we see cancers becoming more common as people age, is that there is more of these steps can take place. So generally the steps that we see is first the cells bypass apoptosis. What this means is that the cell, when it's programmed typically to die, actually can bypass that message and continue to live. So often our cells have a natural lifespan that at the end of that life, then they go through cell destruction and they're replaced with a new healthy cell. Cancer cells first bypass this. Basically, they don't go through cell death. Then they circumvent the need for growth signals, meaning that they become less reliant on the body telling them that they should be going through cell division, and they kind of make up their mind on their own. In addition, they have the ability to escape immunosurveillance, meaning that they can make it so that the immune system doesn't see them. If the immune system doesn't see them and they're growing out of control, that means that there's not really an ability to stop them. They can command their own blood supply. Often we see new blood vessels going to tumors that are growing. They have the ability to basically have their own blood supply that's not dependent on other tissues around them. And then what we typically see is that they may begin to metastasize, meaning breaking apart and starting to spread throughout the body. We would also expect to see that safety net that I said that cells have, which is that tumor suppressor gene, would lose its function. So it no longer has the ability basically to stop all of these earlier steps, and we would expect to see that cancer spread. So what do we do about cancer? So I have kind of a list over here. I'm not going to test you on each of these, but I do want you to think about how long we have been working on cancer therapies. Since 1846, where we first did basically surgery to remove a tumor, and that's one of the best ways that we still have, is basically remove the cells that are cancerous as long as they haven't metastasized. That works fairly well. Often what we do then is that we do surgery, and then in conjunction we do radiation perhaps of that area to make sure that we kill off any of the cells we may have missed during the surgery. We can do chemotherapy, which means that we're giving a drug to the person that basically will hopefully kill off those cancer cells as well. These tend to both be radiation and chemotherapy. Both of these tend to be very hard on the body. More recently, we've started to look at immunotherapy, basically trying to prop up the immune system or to cause the immune system to recognize cancer cells where it otherwise would not. One of the more interesting ways that we've started to think about it, though, is through the use of viruses. We know that there are some viruses that we call oncolytic. So remember, onco meaning cancer, lytic means basically the lysis of. So oncolytic viruses are those viruses that have the ability to find, they're specific for cancer cells, and when they find them, they destroy them. So there's something about cancer cells that oncolytic viruses recognize, and they will basically destroy those particular cells without harming healthy cells. Now, this is kind of a new therapy, and we're still kind of learning about it. But the way that we believe that this works is in four main ways that these oncolytic viruses actually function. The first is that they can infect and replicate within cancer cells. So they specifically target cells that are cancerous, and then they replicate. And as we know, often when we have replicating viruses in a cell, they usually can lyse that cell when they're released. They also sometimes have the ability to do induction of apoptosis. Remember, apoptosis is cell destruction, so cell death. So they can induce that or cause a cell to go through cell death. They also have the ability, in some cases, to induce lysis of that cancer cell, basically burst that cell open and explode, make all those virus particles released, and hopefully they go on to affect other cancerous cells. One of the other things that they can do is complicate the mechanism of those oncoviruses by stimulating the host immune system to actually recognize cancerous cells. So these are kind of the ways that we see oncolytic viruses as a possible virotherapy to cancer. So with that, I'd like to go to the checking in. Please feel free to pause the video and then come back when you're ready to answer the questions with me. So I assume that you either said the answers out loud, or you've written down the answers to these questions. Let's go ahead and go through them. The first question is, what % of cancer is caused by viruses? It's at least 20%. Name one virus known to cause cancer: human papillomavirus, herpes virus, hepatitis B and C. There's also the HTLV and Epstein-Barr virus. Name at least one characteristic of cancer in vivo: evasion of the immune system, uncontrolled cell division, Trying to think of any others off the top of my head. The inability to go through apoptosis, so losing that sense of apoptosis. What is one type of cancer therapy? We have surgery, radiation, chemotherapy, and then the last that is kind of new besides immunotherapy is this idea of virotherapy. So what is one way that virotherapy kills cancerous cells? It can infect and replicate within them. It can cause them to lyse when the virus is released. It has the ability to cause them to go through apoptosis, and also it can alert the immune system to those cells. If you have any questions, please let me know. Otherwise, I think we are done here. Thank you.